All right, welcome back, everybody. Hope everybody had a nice community day on Wednesday. Um, so heads up, we're running into our last bit of content before our exam, which is Wednesday next week. So you have a practice exam posted. We weighted a little bit more heavily some of the more recent concepts so that you can get a bit more practice on those. I'll also post some additional materials for you to practice with over the weekend on Brightspace, so keep a lookout for those. So on Monday, we will be covering a little bit more content and mixing that in with some review. Okay. So feels like it's been a million years, but here is where we left off last time. So what we're looking at here is our hemoglobin oxygen dissociation curve. And what we're thinking about is how do we get oxygen into our red blood cells, onto the hemoglobin? And importantly, this graph is going to help us figure out how much oxygen we actually are able to deliver to our tissues that need it. Okay, so we had talked a bit about partial pressures that we expect to see in our arteries and veins, right? So we can see that here. Those are things that we're paying attention to, right? We know a typical partial pressure of oxygen in a systemic vein is 40. A typical partial pressure of oxygen in a systemic artery is 100. And these correspond to a certain saturation of our hemoglobin molecules, right? So we remember we've got like our 4 little spots for hemoglobin, right? So we can read here on this graph, those 2 points, right? At our partial pressure of 40, we have about 75% saturation of that hemoglobin. And at that partial pressure of 100 in the arteries, right, we're at that like 98.5% saturation. Okay. But we can actually read more from this hemoglobin oxygen dissociation curve, right? So one of the important things that you're going to want to notice is that difference between the saturation in the veins and the arteries, right? So that's why we're looking at these 2 partial pressures and reading these points, is because from a kind of conceptual level, right? Like, why is there a difference in the saturation of oxygen in an artery versus a vein, right? It's because the artery is delivering oxygen to your tissue. The tissue uses that oxygen, and then the vein's just bringing the rest back, right? So that difference between those 2 points tells you how much of that oxygen got unloaded, is the word we use, unloaded from the hemoglobin and delivered to our tissue. Okay. So the shape of this graph is kind of important, and where it shows up on the x-axis is also a little bit important. We're going to see that this is our kind of normal baseline that we reference. But as we go through the next couple of slides, we're going to be talking about right and left shifts. So I want to demonstrate to you what that really means, right? Okay. So if we're going with this as our baseline, right, when we talk about a right shift of this curve, of the hemoglobin oxygen dissociation curve, we shift it to the right, right? All we mean is we're redrawing that line literally a little bit to the right along the x-axis. Okay, so what this shows us, right, is that as we start out with really low saturation of hemoglobin at the very beginning, if we had a partial pressure of 0, right, it's a little bit harder to put oxygen onto that hemoglobin, right? So we describe this as a lower affinity, right? Lower affinity of oxygen for hemoglobin or vice versa, right? Affinity being like how much they like each other, how much they stick to each other, right? So lower is less, right? So as we move to the right with this lower affinity, we do still see this slight uptick usually at the beginning. So this is actually a phenomenon called positive cooperativity. What that meant was once you have one oxygen on hemoglobin, it gets a little bit easier to add more oxygen onto it, right? So that first bit is the positive cooperativity, right? And as we see the partial pressure of oxygen go up, right, we are still going to see the saturation of the hemoglobin go up, the oxygen saturation of the hemoglobin, but not as much and not as quickly as under our kind of baseline. Okay. So if we read this new curve, right, under this rightward shift, we're not going to be thinking about the arteries and veins as having different partial pressures. We're going to see that this is related to other factors. So we're still going to be reading this graph at 40 for the veins and 100 for our arteries. Okay. So now we can see on this graph with the lower affinity of hemoglobin for oxygen, if we read at our vein point and at our artery point, we can now see that there's a really big difference between the amount of oxygen in the arteries and the veins, right? A lot more with this rightward shift than from our baseline, right? So this is more unloading, right? So we have let go of more oxygen at the tissue now. So more unloading under these conditions of lower affinity. So there is a converse point, right? What this means, right, as we described it, right, is it's also hard because of this lower affinity, it's harder to get oxygen onto the hemoglobin. So you might think of that as, harder sounds bad, right? But this is actually kind of good for your body because of this increase in the unloading at the tissue, right? So when we see these effects that create rightward shifts, like say near your muscles that you're actively using, this means you're more effectively able to deliver oxygen to those tissues because it's easier to rip off those oxygens and get them into a different cell, right? Instead of just storing them stuck kind of on the hemoglobin in your red blood cells. So we've got these trade-offs between loading oxygen. So we think of that at the lungs, putting oxygen onto the hemoglobin, and unloading at the tissue. And as we go through the next couple effects that create rightward or leftward shifts, I like to kind of think about an active muscle that you're using, say, on a run or something, right? I like to think of those properties because we're going to see, right here, you can think of this as temperature. One of the things that creates a rightward shift is higher temperature, right? So temperature is going to be one thing. Okay. So if you see an increase in temperature, we'd create a rightward shift with this lower affinity. We could also see leftward shifts, right? So a lower temperature makes it easier to load oxygen, higher affinity, but more difficult to unload. Okay? So I think of my muscles as hot when I'm using them, right? So that's when you'd want a rightward shift. We're going to see that pH has an effect on how oxygen interacts with hemoglobin, the affinity between oxygen and hemoglobin. So you want to think of muscles as being like kind of more acidic when you're using them, when you're breaking down glucose, right? You're producing carbon dioxide as a byproduct. Maybe you're producing lactic acid if we're in a low oxygen environment, right? So rightward shifts for more acidity, so for lower pH. And we see rightward shifts when we're in lower oxygen environments and when we have greater amounts of carbon dioxide. Right? So I like to think of very directly tying this increase in unloading with this lower affinity at active muscle tissue. And then you can think of the reverse potentially if you're thinking about leftward shifts, right? At the lungs, right? A little cooler because you're breathing room temperature air, right? So you might want it to be easier to load oxygen on under those conditions. You can think of examples for yourself for the others. I really just think about what is the rightward shift and then think of the opposite thing for our leftward shifts. Okay. So conceptually, that's how we're looking at these things when we're looking at these hemoglobin oxygen dissociation curves and how they're affecting our oxygen transport in the blood. All right, so temperature is one thing that can affect this affinity of hemoglobin for oxygen, right? But we've got more, right? So pH affects it. We call this the Bohr effect, BOHR, right? So the Bohr effect says that lower pH, so low pH is an acidic environment, is another thing that increases this unloading of oxygen to our tissue. So that would be a rightward shift. And you'd see that big difference in saturation of our hemoglobin and oxygen under these conditions, right? So we're going to think of those active tissues as producing acid. We're thinking of their pH decreasing. We'll talk about this association a little more deeply between carbon dioxide and pH in a little bit. But overall, this pH is causing this rightward shift. So we get more oxygen coming out of those red blood cells and going to tissue that hopefully needs it. Yeah. So carbon dioxide on its own, even if it hasn't affected the pH, also has an effect on this affinity of oxygen hemoglobin, right? So this is our carbamino effect. So think about carbon dioxide, carbamino for that naming there. So the way this works is that when you have carbon dioxide in your blood, it does react with hemoglobin, right? So it doesn't react with the same heme group in exactly the same spot, but we do form what we refer to as carbamino hemoglobin. So there's hemoglobin bound to carbon dioxide. Okay. So when you have this carbamino hemoglobin, it doesn't like oxygen quite as much. So it'll still load some oxygen into those heme groups, but not quite as many as it would if you didn't have a bunch of excess carbon dioxide. So that's a description of lower affinity here, right? So because increases in metabolic activity, aka making ATP, produce carbon dioxide, this is another way that we can get more oxygen to tissue that's actively making and then using energy. This last one requires a bit more squinting, but we're working with the same kind of thing here, right? So when we're thinking about this last effect that we're looking at, this is the effect of 2,3-DPG or 2,3-diphosphoglycerate. Basically, this is a molecule that your body makes under conditions of low oxygen, right? So if you're in like anoxic environments, your red blood cells will make a little bit of this molecule, this 2,3-DPG. So this might be the case if you're anemic. So remember that anemia is when you have this lower capacity to carry oxygen in your blood, right? So you can immediately think, right, if you're not able to carry as much oxygen in your blood, maybe you want something to help out and get more oxygen into tissue, even though you don't have so much oxygen available to you. But we particularly observe this effect in high altitudes. So when you're in a high altitude for a while, your body is in this low oxygen environment, and it starts producing more of this 2,3-BPG. Populations that live at high altitudes kind of all the time sometimes do this even more than other people do. Okay. So the reason you don't see this under conditions that like sea level is that making this 2,3-DPG, so its synthesis, gets inhibited if you have oxyhemoglobin. So if you have a bunch of oxygen bound to hemoglobin, it's going to kind of suppress this effect. Okay. But overall, when you see it, it's decreasing the affinity of hemoglobin for oxygen. So we see this rightward shift of our curve, which means we can let go of oxygen more easily. So we are enhancing our oxygen unloading. And By contrast, right? It's a little bit more difficult to put oxygen on there in the first place. Okay. So those are our big effects when we're thinking about right and leftward shifts. So the temperature, the pH, the carbon dioxide, and whether we're producing a bunch of this 2,3-DPG. But if we want to be, I don't know, complete in understanding what affects how much oxygen we get onto our hemoglobin, we also want to make sure that we give a special shout out to carbon monoxide. So hopefully you all have carbon monoxide detectors in your apartment, because this can seriously impact how much oxygen you can keep in your hemoglobin, but in a kind of different way. Right? So rather than affecting the affinity of hemoglobin for oxygen, so like how much hemoglobin and oxygen like each other and bind to each other, carbon monoxide just like takes up the spots directly where oxygen would bind in, right? So it directly fills up those heme groups, right? And this is because it has a super high affinity for hemoglobin. So hemoglobin really, really likes carbon monoxide. It likes it more than it likes normal oxygen, right? So if you have carbon monoxide in the environment, it's going to be more strongly attracted to those heme groups than your breathable oxygen, your usable oxygen is, right? So we just don't have as much space left for oxygen in the presence of carbon monoxide. So this is why you essentially suffocate. It's not because your lungs aren't actively moving air. It's because all this hemoglobin isn't pulling oxygen around your body. It's just moving a bunch of carbon monoxide around. Okay. So making sure we were all awake. So if we are thinking about this hemoglobin oxygen dissociation curve, we're going to imagine we're going from New Orleans to the top of Mount Evans. So if we're going from a location that's below sea level to a location that's really, really high, over 14,000 feet. So what kind of shift was that, would we see in this case? Okay, let's take a look. Okay, so let's talk this through. Okay, so this is partially an excuse to show you a real graph with real data, but we're making sure that we understand this right for his left shift. So this is about that 2,3 BPG, right? Or DPG, right? Okay. Sometimes we also just call it BPG. I don't know why they have semi-equivalent terms. That's a chemistry specific question, probably. So what we're looking at here is we're looking at this situation where altitude actually affects how much oxygen you have in the atmosphere, right? So usually we have our ballparks for the amount of oxygen we're going to have in our breathable atmosphere. Usually we don't see too many changes, but a time we would see a change is if you go to really high altitudes. You can sometimes feel it, that the air feels thinner, maybe you get lightheaded a bit. People get altitude sickness, right? Part of that is because you're putting yourself in this low oxygen environment. This is why people might like take oxygen tanks with them or something if they're climbing Mount Everest, right? So you're putting yourself in this low oxygen environment. So as long as you give yourself enough time, you'll notice that this curve all the way to the right is showing altitude adapted, right? So you have to give yourself a bit of time to adjust to lower oxygen conditions, right? But 14,000 feet is an amount that you can adjust to physiologically, right? So what we see under these conditions where we have this addition of a molecule that lowers the affinity of hemoglobin for oxygen is this rightward shift, right? So we can see what it looks like if you have absolutely no BPG of this molecule here that affects our oxygen hemoglobin dissociation curve, right? You can see that that's far to the left of a kind of normal amount. So we've got this normal amount of this chemical here in the middle. And under this altitude adapted population, we can see we're even farther to the right here, right? So this would help you out because if we start to read differences, we've got slightly different numbers here in a kind of different unit system at the bottom. So we're not going to look for that 40 and 100 like we usually would. So we're just going to imagine them, right? But if we're imagining it, right, like we could read these two curves at the same spot, right? If we read two spots, let's pick there and there, right? Our amount of unloading increases the farther right we go on this graph here. And that's good. If there's not much oxygen in the atmosphere that you're breathing, you want to make it easier to get whatever oxygen you do have in your blood into your tissue to get used. So in the practice problems or practice documents I'll send you to look at over the weekend, there'll be some tables and stuff about these oxygen hemoglobin dissociation curves so that you can review yourselves. Okay, but that's what we got for oxygen, right? So we're thinking about hemoglobin being the main way we're transporting oxygen through our bloodstream. We know that it's got those 4 heme groups. Those are essentially our spots for oxygen. We've got typical saturations in the veins of about 75%, corresponding to a partial pressure of oxygen in your plasma of about 40. About 98.5% saturation when we look at the arteries, corresponding to that partial pressure of 100 as we move through our systemic circuit. The difference between those saturations is the oxygen that's going into tissue. Okay. So now we're turning to the carbon dioxide side. Right, so when we talked about our partial pressures for carbon dioxide, we know that as we're moving around in an artery, we saw that we expect to see a partial pressure of carbon dioxide of 40. We pass our respiring tissue, our tissue that's making ATP, using ATP. We pick up a bit of excess carbon dioxide there, so our partial pressure goes back up to 46 as we return to the right side of the heart and then go out to the lungs. We breathe out that excess carbon dioxide, drop our partial pressure back down to 40, and swim back around. So that's what we talked about carbon dioxide transport earlier in the week. Now we're thinking about like more of the kind of chemistry, let's say, of that carbon dioxide. So we're thinking a little more nitty-gritty about the transport rather than just our big levels. Okay. So we want to be thinking about how we're exchanging carbon dioxide in our systemic and pulmonary capillaries. We're going to see that just like carbon dioxide affects how much oxygen you can transport in your blood through that carbamino effect, we're similarly going to see that the amount of oxygen you have in your blood also has an effect on how much carbon dioxide you're moving. Okay. So where is this carbon dioxide? Yeah. We got in a couple places. Some of it is directly dissolved in blood, so directly in your plasma. There's more of it in your plasma than there was oxygen, right? So oxygen was very, very little directly dissolved in plasma, like 1-ish%, right? Carbon dioxide, we're up at about like 5 or 6% directly dissolved in blood. We can see we also have a little bit bound to hemoglobin, right? So that's our carbaminohemoglobin that we were just looking at with that carbamino effect, creating rightward shifts of our hemoglobin oxygen dissociation curve. But we can also see that our largest amount of carbon dioxide says that it's dissolved as bicarbonate, right? So we're gonna come back and think about this relationship between carbon dioxide and then hydrogen ions and bicarbonate ions. Okay. So one of the things that we have in our red blood cells and some other places in your body is this enzyme carbonic anhydrase, which we briefly mentioned when we talked about the properties of our red blood cells in our previous unit. All right, so carbonic anhydrase is our enzyme catalyzing the reaction we're looking at here, right? So what we've got is we can put together carbon dioxide in your bloodstream, water in your bloodstream, we've got plenty of that, and turn it here into carbonic acid, which is why we have this name carbonic anhydrase. So this is our carbonic acid. Right. And then not pictured, we have an additional step where often we're going to see, usually we're going to see in the bloodstream, this carbonic acid dissociates into hydrogen ions and bicarbonate ions. Right, so we can tell that those are our ions because they got their little charges on them. Okay, so what happens here when we see increases in carbon dioxide, right? So you got a buildup of carbon dioxide. This means we've got more of our reactants here on the left-hand side of the equation. So it pushes this reaction to the right a bit, right? So as we have more carbon dioxide, we make more carbonic acid, it dissociates, and so we end up with more hydrogen ions and more bicarbonate ions in the blood, right? So we're reading this the way we want to read it, usually left to right here, although this is actually a reversible reaction. Okay. We'll talk about that reversibility when we get to the kidneys. Okay. So we're carrying a lot of that carbon dioxide essentially as extra bicarbonate ions and some hydrogen ions in our plasma because we took that carbon dioxide, we had carbonic anhydrase in the red blood cells, so we turned it into this carbonic acid, and then that carbonic acid dissociated into hydrogen and bicarbonate ions, right? And that's how we transport a fair amount of the carbon dioxide that we see in our bloodstream around. So it wouldn't be noticeable necessarily in that form if you look just at the partial pressure of carbon dioxide. Okay? But we have this kind of buffering effect there. So your blood can store a bit more carbon dioxide than you think. Okay. Separately, what we're looking at here, our final thing, is going to affect that carbon dioxide transport, other than the fact that we can store a little extra as bicarbonate ions using our carbonic anhydrase, is we're going to have this trade-off a little bit between our carbon dioxide and our oxygen. Okay, so we're looking here at some more curves. This curve is less important to memorize than your hemoglobin oxygen dissociation curve, but you do want to kind of understand what we're looking at here, right? So what we're looking at is if you have no oxygen at all, right, you have a relationship between the partial pressure of carbon dioxide in your blood, where more partial pressure of carbon dioxide in your blood, higher total blood carbon dioxide, right? So you're increasing as you increase carbon dioxide. Great. We actually see that in all these lines, right? So the greater partial pressure of carbon dioxide you see in the bloodstream, the greater total amount of carbon dioxide you'll see in the blood. But at any given partial pressure, right, we are going to be able to compare, okay, if we have a partial pressure of carbon dioxide of, let's say 40, right? We're reading it in an artery, okay? If we had no oxygen, even at that lower partial pressure, you'd have a pretty substantial amount of carbon dioxide in the bloodstream. But as soon as you have a bunch of oxygen in that blood as well, this lowers how much carbon dioxide is actually carried by the blood. And this is because kind of the reverse of that effect, carbamino effect, right? So where we have interactions between oxygen and hemoglobin, we also have interactions between carbon dioxide and hemoglobin. So they kind of reversibly affect each other, right? So the greater amount of oxygen you see in the blood, the more it suppresses this sort of carrying capacity of your blood for carbon dioxide. Right? But in both of these effects, while they have different names, right, essentially more oxygen, less carbon dioxide, more carbon dioxide, less oxygen, right? They're affecting each other, which is a little bit what we're looking at here, right? So these next two graphs are showing you some of the effects we see on the unloading of hemoglobin in respiring tissue, so active tissue, metabolically active tissue making ATP, using ATP. And then the next graph, we're going to be looking at the lungs, right? So we're imagining first at our active tissue, right? So our active tissue, that's the spot where we're using up our oxygen, right? So we see this lowered partial pressure of oxygen there. This is where we see that partial pressure of oxygen dropping from 100 down to 40. Okay. So the direct effect here of lowering your oxygen levels is that oxygen goes somewhere, right? So directly, this is the same thing that we're saying when we say that we're increasing our oxygen unloading, right? So lowered partial pressure of oxygen in the blood means it went somewhere. Unloading is that going to tissue piece. Right? We also have these kind of cooperative effects happening at the same time though, right? So one thing that's happening here is the Haldane effect. So when we have that lowered partial pressure of oxygen, right, this means that we have a little higher ability to carry carbon dioxide around in our red blood cells as well. So that's the Haldane effect. And this is why you see that increase in carbon dioxide loading here, right? Now that we're increasing our carbon dioxide loading, some of that carbon dioxide is now on the hemoglobin molecules, right? So that creates our carbamino effect, which is where we have that bound carbon dioxide to hemoglobin having a slightly lower affinity for oxygen, right? Which further increases how much oxygen gets out to your tissue. So that's further increasing the unloading of oxygen because of this lower affinity. So lower affinity, more unloading. Okay. And at the same time as we see oxygen dropping, we are going to almost certainly see our carbon dioxide going up because we're passing by our active tissue. It's getting rid of carbon dioxide as a waste product. And so that's another place that we're getting our increases in carbon dioxide loading, creating this carbamino effect. And at the same time, some of that carbon dioxide, because it's turning to carbonic acid, which is then dissociating into hydrogen ions and bicarbonate, that increase in carbon dioxide is creating a pH change, which creates our Bohr effect. So careful here, higher number of hydrogen ions is the same thing as lower pH, right? And we're not going to look into how we calculate pH right here, right now, but you may have done it before in your chemistry classes. If not, associate these two things together, more hydrogen ions, lower pH, right? So that's describing our Bohr effect here, where our lower pH, our more acidic environments create a lower affinity of hemoglobin for oxygen, helping us release even more oxygen at our tissue. Okay. So questions about this part first before we take a peek at the lungs, which is essentially just going to be the reverse. Okay. So I do recommend like drawing these things out for yourself and practicing explaining them. Once you get them, it'll be easy. Until you get them, it's a bit to work through. Okay. So what we're looking at here is just pointing out that we have the opposite situation in the lungs, right? So in the lungs, this is where we're trying to load oxygen, onto our hemoglobin, right? Because we want to carry more oxygen around through your bloodstream. So we see our partial pressure of oxygen in the blood going up as we're going through the lungs, breathing in air, putting that oxygen onto our hemoglobin. So the word for that is oxygen loading, right? Okay. At the same time, right, we have this Haldane effect. So that's about how oxygen affects how we carry carbon dioxide. So in this case, more oxygen means less carbon dioxide, which means more carbon dioxide unloading, right? Part of the tricky part of learning this is the associating what's more and what's less, this kind of trade-off, but we're unloading carbon dioxide, right? So less carbon dioxide. When we look at the carbamino effect now, right, now we're going to see that we have a higher affinity of oxygen, of hemoglobin for oxygen, right? So this is the opposite. This is that leftward shift, right? So we're going to see this greater loading now in the lungs, right? And as we're passing the lungs, we're also dropping down our carbon dioxide, right? So our direct effect there is affecting carbon dioxide itself. Our indirect effect is that we're affecting pH, but in the opposite direction, right? So we're doing a leftward shift here at the lungs, right? Which means it's going to be easier to get our oxygen onto our hemoglobin. You're going to practice working through those effects, thinking about this hemoglobin oxygen dissociation curve. And when you get kind of lost, try to zoom out and think about what would be useful for your body, because that can kind of give you some tips here. Okay, so we're going to move briefly into our final packet of this unit, which is how we regulate ventilation. Okay. So a reminder, when we're thinking ventilation, right, that's our word for physically moving the air. So we know that when we're ventilating, we're physically moving the air. We're thinking about inspiration, we're thinking about expiration, we're thinking about alveolar pressures, and we're comparing them to our atmospheric pressure. Now that we're thinking about the regulation piece, we're thinking about how does your brain know to shift your breathing rhythm, right? Some of this is voluntary, right? You can deliberately breathe slower or faster, but some of it's not. Most people don't like hyperventilate on purpose, right? So part of this is through homeostasis. Part of this is subconscious, right? Not under your voluntary control. So we're going to have some neurons and some nerves that are involved that are voluntary nerves, but we're also going to have centers in the brainstem that you're not aware of that are adjusting your breathing rate. Okay. So we'll start with the part that's through normal somatic motor neurons. So like named nerves going to skeletal muscle that you might see in lab, right? So our respiratory muscles are skeletal muscles. You have seen them hopefully by this point, right? So we're thinking our diaphragm, and we're thinking our various intercostal muscles between our ribs, right? So since this is skeletal muscle, we know that this is the somatic branch of our nervous system, not our autonomic branch, right? And we know it's under voluntary control, and we're referring to these as motor neurons. And these nerves do have names, and you've certainly seen at least this first one, the phrenic nerve, right? So to tell what nerves we want to associate with inspiration and expiration, we just have to think about what muscle does that, right? So for inspiration, we're trying to make our chest cavity larger. We're making our thorax larger. And that larger space drops down our pressure and lets you suck air in, right? So we're doing that by flattening the diaphragm. Flattening it is actually making it shorter. So that's our diaphragm contraction. So the nerve that goes to the diaphragm and tells it to contract is our phrenic nerve. Our intercostal muscles just have nerves that match the name of the muscle. So they're fairly straightforward. Our external intercostal muscles, so the ones on the outside, looking like hands in your pockets, that help you inspire, pull those ribs up and out, make that chest bigger when you're breathing in. They have a nerve called the external intercostal nerve going to them. Okay. So when you're breathing in, we're mostly just thinking about those two motor neurons, those two somatic nerves going to skeletal muscle. I want to remind us about expiration. So expiration is usually passive. Usually you're just relaxing in order to breathe out, right? Because your relaxed diaphragm shrinks your thoracic cavity. If you relax those external intercostals, your chest collapses in a bit, right? So usually that's all it takes to get enough pressure in your chest in order to breathe out, right? Because you raise it above atmospheric pressure. So usual, typical, easy breathing. You don't need a new nerve. But if you're trying to force it, right? If you're in a situation where maybe your airways are narrowed, right? Maybe you have some reason that breathing becomes more difficult, or maybe you're breathing on purpose more deeply, then you would need your internal intercostal muscles with their internal intercostal nerve, or maybe even some abdominal muscles if you're trying really hard to push out. Okay. So here we can see the activity of our three main nerves that we're tracking, our phrenic nerve and our external intercostal nerve for inspiration, and our internal intercostal nerve for expiration, right? So when you're breathing quietly, only inspiration requires actual nerves to fire, right? So you can see the phrenic nerve and external intercostal nerve are the only things firing when you're breathing easily, nothing going on with your internal intercostal nerve. But if you're breathing hard, right, you can see that we have activity both in those inspiratory muscles and their corresponding nerves. But that's offset with during expiration, we're activating those muscles to shrink our chest a lot more forcefully. So we need that internal intercostal nerve to activate as well, so that we see some tension being generated in that expiratory muscle and lets us push air harder and most probably move more air. So greater lung volumes here, right? So we're going from like our tidal breaths, right? So tidal volumes to maybe that IRV and ERV if we are actively breathing in and out hard. Okay. So that's one piece. That's the mainly voluntary piece. But we do see, and we've got some involuntary pieces, and those are going to have a lot to do with what's going on in your central nervous system, right? So the parts we just talked about in the peripheral nervous system, phrenic nerve, the internal and external intercostal nerves, the peripheral part out in the rest of your body. In the central nervous system, we're going to be tracking inspiratory and expiratory neurons. We're going to be tracking some control centers in the brainstem, so both in the medulla and the pons, as well as something called the central pattern generator. Okay, so fundamentally, an inspiratory neuron is depolarizing during inspiration, right? So that's kind of sending your signal to breathe in. An expiratory neuron is just the reverse, depolarizing during expiration. Depolarizing means basically an action potential. So essentially, we're just saying they're firing when you breathe out. Some neurons are mixed and are kind of halfway between these two. Different respiratory centers are going to have different combinations of these different types of neurons. Okay, so here we're looking at their locations, right? So here we're looking at the brainstem, right? So we know our midbrain is up top, right? Nothing going on there. But we do see we've got some stuff highlighted in the pons, right? So that egg-like kind of swelling. And we've got some things highlighted in the medulla oblongata, leading us down into our spinal cord. Okay. So we're going to focus first on that stuff going on in the medulla here, right? So we're going to be looking at a ventral group and a dorsal group of neurons. So ventral just means belly side. Dorsal, we're thinking dorsal fin on a shark, right? So on the back, right? So we got our VRG on the front, right? So VRG here. And our DRG, that dorsal group, on the back. so posteriorly. Okay. So when we look at these two centers of activity, these two nuclei, they have specifically names. Our VRG includes the nucleus ambiguous, and the dorsal respiratory group includes our nucleus tractus solitarius. I don't expect you to remember those names. But conceptually, what we want to think about is that this ventral group has a mix of both our inspiratory and expiratory neurons. So it's got parts that are controlling breathing in and breathing out. And this dorsal group mainly just has those inspiratory neurons here, right? So we got different types in these different places. And the VRG is really the place you're going to see both. Okay. So this is what you'd see if you were recording the action potentials in the brainstem during inspiration and expiration. So you're going to see this kind of ramping up of action potentials, specifically when you're breathing in, when you're trying to trigger that inspiration. Okay. Popping up to the pons real quick. We've got another RG. This is the pontine respiratory group. Okay. So got some other names too. It's got also sometimes called the pneumotaxic center. It's got all the different types of neurons here, right? So it does a little bit of everything. We've got inspiratory neurons, we've got expiratory neurons, we've got mixed neurons. One of the things we think it does, maybe, that's distinct from what's going on in the DRG and the VRG in the medulla, is we think that maybe it's helping us regulate switching between breathing in and breathing out. So it might be regulating those transition points. There are a fair number of things that we don't actually know about how breathing is regulated from a kind of mechanistic level. Okay. Our most mysterious is the central pattern generator. Maybe there's something new since your textbook was published. I have not dived into the literature anytime recently, but as of your textbook's last update date, the location and mechanism of the central pattern generator were unknown. But conceptually, we think it's establishing our respiratory cycle. One of the hypotheses is that maybe it's doing that kind of the way the heart rhythm is generated. Maybe we have some pacemaker cells, but when they wrote your textbook, nobody had found them yet. So conceptually, something's got to be doing it. We don't really know what. Okay, so the final piece here that we're putting together is that to generate our breathing rhythm, right, we're going to need inputs, essentially. Right, so we're going to need to sense things from your environment. So we will talk on Monday, particularly about these chemoreceptors, right? So chemoreceptors are going to be sensing chemicals. And when we say chemicals, we mean oxygen and carbon dioxide mainly, and really mainly we mean carbon dioxide. So we'll talk about our chemoreceptors. There are also stretch receptors in the lungs. There are receptors that sense irritants like smoke. There are receptors that sense like the position of your body. Those are the proprioceptors. And then we've got things coming from the pons, things coming from your cerebral cortex, so like your voluntary thoughts, all going somewhere to the central pattern generator, which is then sending signals to the DRG and VRG in the medulla to generate your breathing rhythm. Okay, so we will stop there for today. And next time we'll talk a little bit about those chemoreceptors before we conclude this unit.